Half of all the carbon dioxide fixation on Earth is driven by organisms that lack even a basic vascular system. These organisms are algae, simple, autotrophic eukaryotes that dominate the primary production of both marine and freshwater ecosystems. This chart maps the plant kingdom's classification structure. Tracing the descent from Cryptogamia, the non-flowering plants, we arrive at the subkingdom Thallophyta, which encompasses all algal groups. An advanced angiosperm relies on an intricate network of xylem and phloem, anchored by true roots and capped with specialized leaves to transport water and nutrients across long distances. Algae abandon this structural complexity entirely. Their physical form is a thallus, a generalized plant body completely devoid of true roots, stems, or leaves. This physical minimalism is an evolutionary adaptation. By eliminating the need to grow and maintain vascular transport networks, algae dedicate the cellular real estate almost exclusively to photosynthesis and rapid reproduction. To understand algal biology, we must view them as highly optimized biochemical engines built to rapidly process light and carbon in aquatic environments. The internal architecture of a generalized algal cell relies on standard eukaryotic components, but organized for maximum metabolic throughput. The chloroplasts dictate the cell's efficiency. Depending on the species, these organelles assume highly specific geometries, ranging from cup-shaped in Chlamydomonas to tightly coiled ribbon-like structures in Spirogyra. Embedded directly within these chloroplasts are specialized microcompartments called pyrenoids. A pyrenoid features a dense protein core designed specifically to accelerate carbon fixation, which is then surrounded by a thick shell of starch for immediate energy storage. Bounding this entire system is the exterior cell wall. The baseline structure always relies on a rigid inner layer of cellulose, providing structural integrity against aquatic pressure. Inside the chloroplasts, the chemical mechanics of light absorption rely on a specific pigment toolkit. All photosynthetic algae utilize chlorophyll A as their primary catalyst. To maximize efficiency at different aquatic depths, evolution provided accessory pigments. Compounds like chlorophyll B, C, D, fucoxanthin, and phycoerythrin allow specific algal classes to capture exact light wavelengths that penetrate their specific environments. The algal cell operates as a modular chassis. By swapping out pigments and cell wall coatings, the base eukaryotic model adapts to new ecological niches. Algae execute reproduction through 3 distinct biological pathways, deploying each strategy based on environmental conditions. The simplest pathway is vegetative reproduction via fragmentation. The phallus physically breaks apart, and each isolated fragment begins growing into a fully viable clone. In asexual reproduction, the organism releases zoospores. These specialized flagellated spores use whip-like appendages to actively swim through the water column to colonize new territory. Sexual reproduction relies on the physical fusion of 2 distinct gametes to mix genetic material. This gamete fusion takes 3 forms: isogamous, where both gametes are identical in size, anisogamous, where the gametes differ in size, and oogamous, which pairs a large, static egg with a small, highly motile sperm. This flowchart maps the haplontic life cycle, a reproductive strategy that reveals a central paradox of algal biology by tracking the haploid N and diploid 2N states. Following the main loop of the cycle, we see that the dominant, free-living, photosynthetic plant body is entirely haploid. This is the gametophyte, and its primary function is to generate haploid gametes. Through syngamy, the biological term for fertilization, 2 gametes fuse to create a diploid zygote, entering the 2N state. In most algae, there is no free-living diploid plant body. The entire sporophytic generation is represented solely by this single-celled temporary zygote. The zygote undergoes immediate meiosis, splitting back into haploid spores that restart the dominant gametophyte loop. There are strict exceptions to this rule. Species like Ectocarpus and Polysiphonia employ a haplodiplontic cycle with alternating multicellular phases, while Fucus relies on a diplontic cycle similar to higher animals. By utilizing the haplontic cycle, the vast majority of algae bypass the energy-intensive diploid growth phases seen in complex land plants, prioritizing explosive generation turnover. This comparative matrix organizes the three major algal classes by cataloging strict variations in pigments, stored food, cell wall composition, and flagella structure. The first column defines the Chlorophyceae, or the green algae. Their biochemical profile is defined by chlorophyll A and B. They store energy as starch, utilize rigid cell walls built of cellulose and pectose, and feature motile stages with apically inserted flagella. This combination of pigments and starch storage proves their evolutionary significance. Green algae are the direct biological ancestors of all higher land plants. The second column details the Phaeophyceae, the brown algae, which include the marine giants of the plant kingdom. They operate using chlorophyll A and C, but their distinct color comes from a heavy concentration of the accessory pigment fucoxanthin. Their internal chemistry is modified to store complex carbohydrates like mannitol and laminarin. Externally, their cellulosic walls are coated in a thick, gelatinous layer of algin. Despite remaining a thallus lacking true vascular tissue, brown algae achieve massive structural differentiation, organizing into a root-like holdfast, stalk-like stipe, and photosynthetic frond. By incorporating fucoxanthin and swapping starch for laminarin, the algal chassis scales up from microscopic green dots to oceanic kelp forests reaching 100 meters. The final column identifies the Rhodophyceae, the red algae, highly specialized for deep-water marine environments. Their pigment profile relies on chlorophyll A and D, driven by heavy concentrations of their defining compound, R-phycoerythrin. R-phycoerythrin actively absorbs blue light. Because blue wavelengths possess the highest energy, they are the only light spectrum capable of penetrating into deep ocean trenches, allowing red algae to photosynthesize where other plants cannot survive. Red algae store their energy as Floridian starch. This specific carbohydrate is structurally identical to the amylopectin and glycogen stored in human muscle tissue. Their reproductive strategy is entirely static. At no point in their life cycle do red algae produce flagellated or motile cells. They rely entirely on water currents for gamete dispersion. With the matrix complete, the taxonomic logic of algae becomes clear. The visible color of an alga is a direct readout of its evolutionary depth gauge and metabolic survival strategy. Understanding the rigid taxonomy of these organisms allows human industry to extract specific biochemical tools from each class. At the macroecological scale, the sheer volume of algal photosynthesis drives at least 50% of the total carbon dioxide fixation on the planet, sustaining the dissolved oxygen levels required by marine food webs. Commercially, humans harvest algae for their hydrocolloids. Hydrocolloids are complex, water-binding polymers that algae engineer to prevent themselves from drying out during turbulent low tides. The specific hydrocolloid extracted depends on the class. Algin is harvested exclusively from the cell walls of brown algae, while carrageen is processed from red algae. A third major product, agar, is extracted directly from the red algae species Gelidium and Gracilaria. Agar provides the structural backbone for commercial jellies and serves as the solidifying agent for culture mediums. It is the chemical foundation that makes all modern microbiological lab work possible. Certain marine brown algae, such as laminaria, concentrate heavy trace minerals from seawater, making them a dense, highly harvestable source of iodine for human nutrition. Unicellular green algae, specifically chlorella and spirulina, are cultivated as single-cell protein, or SCP. SCP refers to edible microbes grown rapidly in industrial bioreactors to yield massive amounts of protein. The metabolic efficiency of these specific green algae is so high that they are heavily utilized as concentrated, low-weight food supplements for space travelers. By analyzing the thalloid structure, haplontic cycles, and precise pigment matrices of algae, we recognize them as the biological engines that sustain the aquatic biosphere and anchor the evolutionary history of the plant kingdom.